Bacterial flora of remote tribes people carries antibiotic resistance genes. Date: April 17, 2015. Source: Washington University in St. Louis. Summary: Scientists have found antibiotic resistance genes in the bacterial flora of a South American tribe that never before had been exposed to antibiotic drugs. The findings suggest that bacteria in the human body have had the ability to resist antibiotics since long before such drugs were ever used to treat disease. These are huts in an isolated village inhabited by Yanomami Amerindians in a remote, mountainous area in southern Venezuela. Members of the tribe were isolated from the modern world and had never been exposed to antibiotic drugs. But the bacteria on their skin and in their mouths and intestines still had antibiotic resistance genes. Credit: Oscar Larkin. Scientists have found antibiotic resistance genes in the bacterial flora of a South American tribe that never before had been exposed to antibiotic drugs. The findings suggest that bacteria in the human body have had the ability to resist antibiotics since long before such drugs were ever used to treat disease. The research stems from the 2009 discovery of a tribe of Yanomami Amerindians in a remote mountainous area in southern Venezuela. Largely because the tribe had been isolated from other societies for more than 11,000 years, its members were found to have among the most diverse collections of bacteria recorded in humans. Within that plethora of bacteria, though, the researchers have identified genes wired to resist antibiotics. The study, published April 17 in Science Advances, reports that the microbial populations on the skin and in the mouths and intestines of the Yanomami tribes people were much more diverse than those found in people from the United States and Europe. The multi-center research was conducted by scientists at New York University School of Medicine, Washington University School of Medicine in St. Louis, the Venezuelan Institute of Scientific Research and other institutions. This was an ideal opportunity to study how the connections between microbes and humans evolve when free of modern society's influences, said Gautam Dantas, Ph.D., Associate Professor of Pathology and Immunology at Washington University and one of the study's authors. Such influences include international travel and exposure to antibiotics. Intriguingly, in Dantas' lab, graduate student Erica Pesson searched for and found antibiotic resistance genes in bacteria on the skin and in the mouths and intestines of tribe members long isolated from such outside influences. These people had no exposure to modern antibiotics. Their only potential intake of antibiotics could be through the accidental ingestion of soil bacteria that make naturally occurring versions of these drugs, Pesson said. Yet we were able to identify several genes in bacteria from their fecal and oral samples that deactivate natural, semi-synthetic and synthetic drugs. Thousands of years before people began using antibiotics to fight infections, so bacteria began producing natural antibiotics to kill competitors. Similarly, microbes evolved defenses to protect themselves from the antibiotics their bacterial competitors would make, likely by acquiring resistance genes from the producers themselves through a process known as horizontal gene transfer. In recent years, the abundance of antibiotics in medicine and agriculture has accelerated this process, stimulating the development and spread of genes that help bacteria survive exposure to antibiotics. Consequently, strains of human disease that are much harder to treat have emerged. We have already run out of drugs to treat some types of multi-drug resistant infections, many of which can be lethal, raising the bleak prospect of a post-antibiotic era, Dantas said. Scientists don't really know whether the diversity of specific bacteria improves or harms health, Dantas said, but added that the microbiomes of people in industrialized countries are about 40% less diverse than what was found in the tribes people never exposed to antibiotics. Our results bolster a growing body of data suggesting a link between, on one hand, decreased bacterial diversity, industrialized diets and modern antibiotics, and on the other, immunological and metabolic diseases, such as obesity, asthma, allergies and diabetes. which have dramatically increased since the 1970s, said Maria Dominguez-Bello, Ph.D., Associate Professor of Medicine at New York University Langone Medical Center, and senior author of the study. We believe there is something occurring in the environment during the past 30 years that has been driving these diseases, and we think the microbiome could be involved. Dominguez-Bello said the research suggests a link between modern antibiotics, diets in industrialized parts of the world, and a greatly reduced diversity in the human microbiome, the trillions of bacteria that live in and on the body, and that are increasingly being recognized as vital to good health. The vast majority of human microbiome studies are focused on Western populations, so access to people unexposed to antibiotics and processed diets may shed light on how the human microbiome has changed in response to modern culture, and may point to therapies that can address disease-causing imbalances in the microbiome. In the current study, when the researchers exposed cultured bacterial species from the tribe to 23 different antibiotics, the drugs were able to kill all of the bacteria. However, the scientists suspected that these susceptible bacteria might carry silent antibiotic resistance genes that could be activated upon exposure to antibiotics. They tested for such activation, and the tests confirmed their suspicions. The bacterial samples contain many antibiotic resistance genes that can fend off many modern antibiotics. These genes may turn on in response to antibiotic exposure. However, we know that easily cultured bacteria represent less than 1% of the human microbiota, and we wanted to know more about potential resistance in the uncultured majority of microbes, Dantas said. So the researchers applied the same method, called functional metagenomics, to identify functional antibiotic resistance genes from Yanomami fecal and oral samples without any prior culturing. From that experiment they were able to identify nearly 30 additional resistance genes. Many of these genes deactivated natural antibiotics, but the scientists also found multiple genes that could resist semi-synthetic and synthetic antibiotics. These include, for example, third and fourth generation cephalosporins, which are drugs we try to reserve to fight some of the worst infections, said Dantas. It was alarming to find genes from the tribespeople that would deactivate these modern synthetic drugs. As for how bacteria could resist drugs that such microbes never before had encountered, the researchers point to the possibility of cross-resistance, when genes that resist natural antibiotics also have the ability to resist related synthetic antibiotics. We've seen resistance emerge in the clinic to every new class of antibiotics, and this appears to be because resistance mechanisms are a natural feature of most bacteria, and are just waiting to be activated or acquired with exposure to antibiotics, Dantas said.